A microarray, or DNA chip, is a spatial array of oligonucleotide probes, closely arranged on a small, solid support surface. The oligonucleotide probes, representing nucleotide sequences in known genes, are synthesized on the solid surface in such a way that both the position and nucleotide sequence of each probe are known. Microarrays can be used to identify organisms and specific genes by hybridization of DNA from the organism to the oligonucleotide probes in the microarray. There are several ways to make a microarray. The one described here involves a methodology adapted from computer chip technology. A piece of glass, about 1.5 by 1.5 centimeters, is covered with a mask containing holes corresponding to the sites of attachment of the desired nucleotides. The first nucleotide to be attached is added and made to bind by a light activation reaction. The mask is then replaced by another mask which exposes sites for the addition of the next nucleotide. This process is repeated until all of the oligonucleotide probes, each with a known sequence and position, is synthesized. Although only 5 nucleotides are shown in the oligonucleotides of this animation, there are usually 15 to 25 nucleotides in length in the microarray. The DNA from the organism being studied is isolated, fragmented, and tagged with a fluorescent dye. The DNA fragments are then incubated with the chip. DNA from the organisms that is complementary to the DNA in the microarray bind together, and the unbound DNA is washed away. The surface of the microarray is then scanned with a laser beam, and the data obtained is used to produce a visible image. Color intensity indicates the extent of hybridization to the different probes. Over 200,000 probes can be constructed on a single microarray. With this technology, any desired sequence of nucleotides can be constructed to form a probe. The presence of genes for antibiotic resistance and other traits can easily be determined, and gene expression can be monitored by hybridizing cDNA obtained from messenger RNA.